Good morning, everyone, and welcome to this webinar, ELISA and Beyond. My name is Teresa, and I'm here today with my colleague Rebecca. Together, we will provide you some information about ELISA and other immunoassays. First of all, we will introduce ourselves. Hi again, my name is Teresa, and I'm working as a bioresearch field application scientist since January of 2021. Previously to join the company, I did my PhD about nutrition and type 2 diabetes. What do I normally do on my free time? Well, I love to try new food, so consider myself as a foodie, and I also love reading books. In addition to that, I enjoy traveling, doing yoga, and I have just started dancing. Now I will pass on my colleague Rebecca.
Thank you, Teresa. Good morning, everyone. My name is Rebecca, and I'm a field application scientist for molecular devices. My background is in neuroscience, where I studied in University College Dublin. I joined molecular devices about a year ago and currently cover Ireland and the Nordics. Some of my hobbies include eating, as you can see here on the left, also running and tennis in my spare time, and I am an avid animal lover. Here's a picture of my little dog Willow on the right. So to start off, here is a brief look at what we will cover today. We'll look at what is an ELISA, its three base detection modes, and then finally moving on to some more advanced technology such as HTRF and alpha technology. So first off, what is an ELISA? ELISAs are a type of immunoassay that are commonly used to quantify levels of a specific target within a sample. Samples include serum, plasma, saliva, which you'll be familiar with the COVID tests, and also with pregnancy tests, as the example here is urine. There are four main types of ELISAs. The first one here is a direct ELISA. An antigen or sample is immobilized directly on the plate, and a conjugated detection antibody binds to the target protein. The substrate is then added in suspension, producing a signal that is directly proportional to the amount of analyte in the sample. These are used when assessing antibody affinity and specificity, and also when you want to investigate blocking or inhibitory interactions. An indirect ELISA is similar to a direct ELISA in that an antigen is immobilized again on a plate, but it includes a conjugated secondary antibody, shown here in blue, directed against the host species of the primary antibody, shown here in green. The suspended substrate then again produces a signal directly proportional to the amount of analyte or antigen bound in the well. So these are used when measuring endogenous antibodies. Moving on to a sandwich ELISA, which is the most common type of ELISA. Two specific antibodies are used to sandwich the antigen, commonly referred to as matched antibody pairs. A substrate then in suspension again is added and produces a signal that is directly proportional to the amount of antigen present in the sample. These are usually used when determining analyte concentration in a biological sample. And lastly, we have the competitive ELISAs, which are commonly used for small molecules, when the protein of interest is too small to efficiently sandwich with two antibodies. So here the antigen competes with the reference antigen for binding of a specific amount of labeled antibody. The reference is pre-coded on a multi-well plate, and the sample is pre-incubated with the labeled antibody and added then to the wells. So depending on the amount of antigen in the sample, more or less free antibodies will be available to bind to the reference. Depending on the amount of antigen in the sample, more or less free antibodies will be available to bind the reference antigen. This means that the more antigen there is in the sample, the less reference antigen will be detected and the weaker the signal. These are usually used when determining concentrations of small molecules or hormones. So here is a table showing which ELISA to choose from and their advantages and disadvantages. For direct ELISAs, they're fast and simple. However, they can be less specific as it only uses one antibody and has potential for high background if all proteins from a sample are immobilized in the well. For indirect, amplification arises using the secondary antibody. There is, however, potential for cross-reactivity caused by the secondary antibody. For a sandwich ELISA, has the highest specificity and compatible with small matrices. However, the protocols can be long and challenging to develop. For a competitive ELISA, they have the ability to quantitate small molecules. However, again, they can be less specific since you're only using one antibody and requires the conjugate antigen. The illustration above shows a workflow for a typical sandwich ELISA. You can see here that there is 8 steps. You have your 96 to 3L microplates where your antibody binds to the well, then the addition of your sample, and then here shown in red you have your 2 washing steps, which can be very time consuming as it can take over an hour for these washing procedures. You usually wash the microplate 3 times in each wash steps, which can be a total of 6 washes. Then here with 7 and 8, the microplate reader is used to detect the colour change produced when the target antigen is present. And then lastly, you have your software, which is used to plot your standard curves and calculate results. This workflow can also be fully automated. We all know too well that manual microplate washing with pipettes can be very painful and laborious, quite like this picture here on the left. I can remember when I was doing my ELISAs in the lab, carrying out the washing steps can be very, very laborious. However, with the use of a microplate washer, you can reduce these time-consuming steps by washing the wells without the need for pipettes, similar here to the picture on the right. It would have made my life a lot easier in the lab. By using a microplate washer for the washing steps 3 and 5, you will achieve efficient cell washing with minimum cell loss and optimal assay performance. It allows for precise dispense and aspiration control, allowing processing without cell disruption. The washer does not require any external pumps or computer to operate, minimizing bench space. The washer is easy to integrate with the latest automation tools, and it is compatible with the Molecular Devices Stack Max and many robotic plate handlers. Now we will move on to Absorbance and ELISA. To start off, we will ask the question, what is absorbance? Well, it is when a molecule absorbs light at specific wavelengths and efficiencies. Optical density donates to the amount of light absorbed by the solution. You can see on the left picture below where absorbance follows the Beer-Lambert law, where OD is directly proportional to the concentration of the sample being tested. On the right is a picture giving a present representation of the optics within a microplate reader and how the OD read is achieved. You have your light source here above, which is usually LED or xenon flash lamp, then your monochromator or filter, your sample then below, and your photodiode, a forward detection. Here are some absorbance applications below. And it's shown in red are your ELISA and immunoassays. These can be read in single endpoint, in kinetic, well scan, and so on. Below also you have the ability for microvolume applications, which you can use the spectrograph that can quantify as little as 2 to 4 microliters. of a sample. So the next slide here is very fitting with Irish culture. If you can't tell from my accent, this is where I'm from. So a lot of people in my country will be very happy with this research. This is an example of how gluten was quantified in beer using an ELISA method. I will just briefly synopsize the study. There has been recent rise in the prevalence of celiac disease, and monitoring gluten levels in food and beverages has become increasingly important as more people strive to avoid gluten. The Ridoscreen gliadin competitive ELISA can detect small peptide fragments using a standard material, hydrolysate of wheat, rye, and barley. The OR5 antibody used in this kit recognizes potential toxic peptide sequences of gliadins from wheat and prolamins from rye and barley. The competitive ELISA provides a quantitative solution for gluten detection in beer. The hands-on time required to run this assay is reduced by using a multi-wash washer to automate steps. Detection of absorbance with the SpectraMax Abs Plus reader and the calculations of results using our SoftMax Pro software further simplify the ELISA workflow and flag any result that warrant further attention. So now we all can enjoy a beer at the pub. This is the family of single-mode readers for molecular devices which are compatible with Absorbance. The SpectraMax Quick Drop, the Emax Plus, the VersaMax, and the SpectraMax Abs. Now we will move on to Fluorescence and ELISA. So again, we'll look at the principle of fluorescent intensity. It is when a molecule absorbs energy and it is transformed from a ground to an excited state, shown here below. It then releases the energy in the form of light of a lower intensity and longer emission wavelength. Due to the energy dissipation during the excited state lifetime, the emission energy of a photon is lower. It therefore has a longer wavelength than the excitation, as you are losing some of the energy in the form of heat. The difference in energy or wavelength is called the Stokes shift. The Stokes shift is fundamental to the sensitivity of fluorescent techniques because it allows emission photons to be detected against a low background, isolated from the excitation photons. Depending on your fluorophore, the Stokes shift can flux in nanometers. A microplate reader with fluorescence intensity detection uses a light source, which is a xenon flash lamp or LED, to excite the fluorophore. The wavelength required to excite the sample can be selected using either a filter of a specific wavelength or a monochromator tuned to the required wavelength. The fluorophore then emits light of a different wavelength selected by a second filter or monochromator. This emitted fluorescence is detected by a photomultiplier tube, and the fluorescence intensity of a sample is expressed as relative fluorescent units. With our reader, there is little optimization necessary due to the unique automatic PMT. When selected, the software will automatically normalize the data within each well, so it is independent of the PMT gain selected. Therefore, the data from each well and plate can be comparable. You can also have the option of reading from the bottom, which is advantageous for coated plates or cell-placed assays where the signal is coming from the bottom of the well. A lot of molecules may absorb light, however a lot less will emit light. Therefore, fluorescence can have less background compared to absorbance. So fluorescence applications are similar to absorbance, but they will have a lower background and therefore improved sensitivity. Here are some of the fluorescence applications and where fluorescence ELISAs are highlighted in red. This is a schematic workflow depicting sequential molecular binding events of a sandwich fluorescent ELISA. Within each well, the capture antibody is absorbed on the reactive surface, followed by surface passivation by a blocking buffer to reduce any non-specific antigen binding. Upon target binding to capture the antibody, alkaline phosphatase tag detection antibody specific to the antigen is added. Addition of fluorescent substrate activated by AAP generates a detectable fluorescent signal, indicating successful binding events. Here is an example of the catch point simple step ELISA kits, which have been developed using a fluorescent substrate to provide improved linearity over an extended dynamic range, providing better quantification of both the lower and upper ends of the curve. Both an absorbance and fluorescent ELISA kit were plotted on the same graph for comparison, shown on the left here. On the y-axis are logged values. On the left-hand side shows a log OD standard curve for a basic colorimetric ELISA kit. You can see that the lower part of the curve to the upper part are only approximately a 1.5-fold range in OD, in comparison to the right-hand side, which shows an approximate 3-log-fold in RFU values for the catch point ELISA kit, depicting a greater increase in dynamic range for the assay. You can also see in the colorimetric ELISA kit that the linearity starts to drop off at around 1,000 picomole per mil on the x-axis, whereas the catch point ELISA is linear up to 10,000 picomole, greatly increasing the detection limit of the human ICAM-1. Some of the other additional benefits include the extended dynamic range, which I mentioned, stable signal for readout flexibility, no compromise in sensitivity, it's only a one wash with only 90 minutes, and high quality antibodies from batch to batch provide consistency. The single mode reader for molecular devices, which is compatible with fluorescence, is the Gemini. Now we will have a look into luminescence and ELISAs. Firstly, again, we will look at the principle of luminescence. It is the emission of light by a substance as a result of a chemical or enzymatic reaction. No excitation component is needed. However, it needs a dedicated detector, which is a photomultiplier tube in photocounting, to ensure assay sensitivity. Below is an example of a chemiluminescent reaction. On the left-hand side of the reaction, you have your reactants, which are catalyzed by firefly luciferase, and in turn producing products and light. There are two principal chemiluminescent reactions, which are shown here in the figure below. The graph on the left shows a flash reaction. It causes a flash of light. As you can see here in the graph in red, the reaction is extremely rapid, and it generally needs injectors as the time period is within seconds, shown below here on the x-axis. The graph on the right, shown here in green, is a glow reaction. As you can see, the RLU values increase slowly over a period of time, and they remain stable for usually minutes or hours. One of the advantages of luminescence is that most molecules and atoms either absorb or fluoresce. Not many luminesce. Therefore, the sensitivity of luminescence is the greatest. Here are some of the luminescent applications where the luminescence ELISA is highlighted here in red. With COVID, I had a lot of spare time and binge watched a lot of crime series. And I always wondered why they would turn off the lights during crime scene investigations and see a blue glow spot in certain places. So they actually use an organic compound called luminol, which exhibits a chemiluminescence with a blue glow when mixed with an appropriate oxidizing agent. Therefore, forensic investigators use luminol to detect trace amounts of blood at crime scenes, as it reacts with the iron in the hemoglobin. So this is just showing the compatible luminescence reader, the spectrometer from molecular devices. This table then shows a brief overview of the detection strategies for ELISAs. All modes use the same enzyme probes, which are horseradish peroxidase and alkaline phosphatase. However, from absorbance to fluorescence, there is an increase in sensitivity. And also from fluorescence to luminescence, there's a 1,000-fold in sensitivity. Below here are some of the advantages to take into consideration when picking which read mode to use. For absorbance, there is direct visualization of the OD, and there's high reproducibility between plates. With fluorescence, you're getting an increase in dynamic range. However, with luminescence, it is the most sensitive detection strategy, and again, you have a wide dynamic range. When choosing which plates to use, it is recommended that a clear plate is used for absorbance, and if you're in the UV range, below 3 nanometer, you use a UV plate. With fluorescence, it is recommended to use a black microplate, and for luminescence, it is recommended to use a white microplate. Our multimode readers shown here are compatible with all three detection modes explained: absorbance, fluorescence, and luminescence. Now I will pass the presentation on to Teresa, where she will explain further ELISA detection technologies.
Thanks, Rebecca, for giving us a detailed explanation about how an ELISA is working. Now, I would like to move to the ELISA workflow again and pointing out some challenges from this technology. The first challenge to mention is the washing steps, increasing assay length and time. Also, having a fixed plate format, which is commonly used a 96 well plate, which makes it difficult to increase the throughput. Limited sensitivity and dynamic range, and finally, short signal stability, which is normally around 1 hour, except for luminescence ELISAs. As you can see in the picture, 8 steps are needed in order to complete the whole ELISA workflow. So now the question to ask is whether do we have other type of immunoassays which can be alternative to ELISA assays, improving the sensitivity and dynamic range. Also, where no washing steps are required, increasing the signal stability and possibility of changing to 384 well plates, increasing the throughput. This alternative can be HTRF and alpha technology. Despite their fancy names and that you might not be familiarized with them, they are fluorescence and luminescence-based technologies. Let us start with HTRF, which stands for Homogeneous Time Resolved Fluorescence. As you can see in this workflow, in this case, no washing steps are needed. Also, the assay time is shorter, only 1 or 2 hours, in comparison to an ELISA assay, which is between 4 hours and 24 hours. Also, the signal stability is from 24 hours up to days, and finally, the dynamic range is up to 4 logs. The increased signal stability will allow you to go home and read at the following morning, in case you finish an essay late in the afternoon. Finally, this workflow is only 4 steps, in comparison to the 8 steps in the case of the ELISA assay. But now the question is, how does HTRF work? As I have just mentioned, HTRF stands for Homogeneous Time Resolved Fluorescence and is a TR-FRET technology developed by SysBio. But what does TR and FRET means? Well, let's start with FRET. From one side, FRET means fluorescence resonance energy transfer. Basically, there are two fluorophores, a donor and an acceptor, which can be, for example, protein, DNA. These two molecules are conjugated to two fluorophores, the donor and the acceptor. The peculiarity of these two pairs of fluorophores is that in close proximity, the emission of the donor can excite the acceptor. In the other side, TR means time resolved and is a base in fluorophore with long-lived fluorescence lifetime. I will explain a bit more in depth in the following slides. So the standard fluorophore like fluorescent has a short-lived fluorescence emission in the range of nanoseconds, as you can see in the graph in the orange line. Excitation and emission light are measured with a really small delay, producing a high background signal. The background signal is normally coming from the solution. Other fluorophores such as europium or terbium emits long-lived fluorescence, which lasts for milliseconds. In this case, excitation and emission are measured with a delay, so the signal-noise ratio is increased, increasing also the sensitivity of the assay. Because of the high background might interfere with the signal, we need to use filters in order to achieve the sensitivity needed for the HTRF assays. Therefore, as we can see in this picture, the plate reader is provided with an excitation filter for the excitation of the donor and with two emissions filters, one for the emission light of the donor and one for the emission light of the acceptor. Because the donor will excite the acceptor, only one excitation filter is needed, in this case for the donor. But how is this technology being measured in our plate reader and software? Well, as mentioned at the beginning, some important and differential parameter from this technology is a delay time between the excitation of the sample and the collection of the signal. This parameter can be changed in our software in order to set up your assay. In general, you will need more optimization than for the ELISAs. As you can see, more parameters need to be optimized in order to set up your assay, and we can offer you some already optimized protocol from our multimode readers. Regarding our multimode reader, here are shown two of them, which are compatible with HTRF measurements. These instruments are combined with dedicated filters in order to fulfill your HTRF specification. In the case of the ID5, a filter slider is combined, and in the case of the I3X, a cartridge is being added. As summary, here are shown some key features from HTRF like long-lived fluorophore like europium and terbium, high signal stability, homogeneous assays where no washing is needed, able to miniaturize, high sensitivity due to low background, and data normalization, allowing direct data comparison. In this technology, a ratiometric ratio between the fluorescence from the acceptor and the donor is being calculated. Now let's move to alpha technology. As we can see in the workflow, no washing steps are required. The assay time is shorter, so only between 1 and 2 hours. The sensitivity is improved, reaching 1 picogram per milliliter, and the dynamic range is 5 lux. An additional advantage of the alpha technology in comparison to the ELISA is the capacity of multiplexing, where up to 3 different analytes can be detected at the same time in comparison to ELISAs, where only one analyte is detected. Also, only 5 steps are needed in comparison to the 8 steps for ELISA. So, how is alpha technology working? Alpha technology have been developed by Perkin and Elma and is a non-radioactive assay. Alpha stands for Amplified Luminescence Proximity Homogeneous Assay. It is a bit-bed assay in which the donor and acceptor bits are conjugated to biomolecules, for example, receptor ligands or antibody antigen. When in close proximity, a luminescence fluorescent signal is produced. Now, I will move to the next slide where I will shortly explain in more details how this technology works. First of all, a laser will excite at 680 nanometer a molecule from the donor bit. This produces the conversion of the oxygen into a singlet oxygen that reacts with different chemiluminescers of the acceptor bits, activating fluorophores that emits light. Depending on the type of fluorophore that's been activated, there are three main types of technologies: alpha screen, AlpaLISA, and AlpaPlex. Here I'm showing the molecules in the acceptor bits that they are activated and the signal cascades that is being generated, producing light at different wavelengths. For example, for alpha screen, the signal is from 520 to 620 nanometer. For AlpaLISA at 650 nanometer due to europium, and finally for AlpaPlex at 545 and 645 nanometer due to terbium and samarium. In order to be able to measure alpha technology, one of our multimode reader, the SpectraMass i3X, together with a filter cartridge, is compatible for measuring alpha screen and AlpaLISA. Some benefit from these cartridges is the use of a strong light source, a laser diode, and also the use of an excitation and emission filters. As you can see, having a look at the wavelength of the filters, the excitation wavelength is higher than the emission wavelength. We can see in this graph, the alpha screen emission is broad, so the emission filter we use is 100 nanometer bandwidth, passing as much light as possible and increasing the sensitivity of the assay. This is the reason why we can measure also the AlpaLISA technology, so the emission filter is catching the signal at 620 nanometer. However, alpha technology can also have some possible limitation. So it is known that the signal produced is really bright, generating a glowing effect and increasing the crosstalk. As you can see here in this picture, sometimes the instrument is catching light from adjacent well. This effect is called crosstalk and will depend on instrument design. Some possible solutions for that are a light blocking tube available in the alpha screen cartridge and the interlace reading available in our software. I will shortly explain a bit more in details. So as you can see in the picture, the cartridge is provided with a light blocking tube which will move out during the read and situated on top of the well in order to block away any light from the neighboring wells, reducing the crosstalk. An additional solution for reducing crosstalk is the interlace reading in our software. The instrument is reading the plate as a chessboard. As you can see in the lower picture, the software is reading A1, A3, A5, and so on, and then reading again the plate, in this case the wells A2, A4, A6, and so on. This software feature is helping to reduce crosstalk in addition to the light blocking tube. Some benefits from the interlace reading are a decrease of crosstalk by 13%, lower CV values, and in general, better curves. Now I will show you some data from an alpha screen assay run together with my supervisor in the lab. In these two graphs, we can see the effect of the interlace reading on versus off in three different samples. So as you can see in the lower part of the graph, the CV values are reduced, and in general we can observe better curves when the interlace reading was on in comparison than when the interlace reading was off. As summary, alpha technology has the following advantages: high sensitivity assay, low background, high signal background ratio, able to miniaturize, a cost-effective assay, versatility, flexibility, and low affinity to high affinity interactions. Thanks a lot for your time, and please let us know if you have any questions.